Molecular genetics is a broad subject with an extensive list of topics that led to many interesting scientific discoveries. Dr. Soy and Dr. Galabuzzi decided to analyze in depth and understand the complexities of viroids and prions.
To begin, viroids are composed of small, non-protein coating infection stranded RNA molecules. Viroids are pathogens, which are microorganisms that cause disease within plants. Their targets are generally higher plants. Viroids are found in extracellular membrane and may or may not contain a phospholipid protein called an envelope, and a protein coating surrounding its nucleic acid called a capsid. Since viroids are self-replicating, they are said to resemble viruses due to their similar structures and functions. Examples of viroids include spindle tuber disease in potatoes, which are slowly transmitting into tomato plants, avocado sun blotch viroid, peach latent mosaic viroid, and pear blister canker viroid.
Prions are also pathogens, implying that they are small infectious particles as well. But instead of being composed of RNA molecules, prions are proteinaceous, meaning that they are containing, resembling, and/or even being a protein. They themselves do not contain nucleic acid, but however contain a prion protein, PRP. Prion diseases can be represented as genetic infection or sporadic disorders, and involve the modification of PRP and resist inactivation by procedures which involve the modification of nucleic acids. Examples include mad cow disease, fatal dementia known as Creutzfeldt dementia, neurological diseases, croup, and in sheep, scrappy.
Viroids, which can be assumed to function like a virus just from their similar names, rely on their host for replication because they lack the protein coding capacity that viruses have. This was reported by Theodore Diner in 1971. In 1965, Diner and William B. Raymer discovered that spindle tuber disease within potatoes was not due to a virus but a viroid. Viroids are able to quickly move from cell to cell as a virus would, causing disease quickly in plants. Viroids have circular or ring-like structure that is comparable to the genetic material in bacteria called plasmids. However, viroids are dependent on RNA polymerase to infect the transcription of protein in order to replicate themselves. This is called the rolling circle mechanism. RNA polymerase, which is normally associated with the synthesis of mRNA, is used to catalyze RNA strands using a viroid template to transcribe the RNA template. RNA polymerase, with the assistance of the host proteins, then starts transcription of RNA and implies the viroid promoter. The process is similar to the use of recombinant DNA to place non-virulent segments in the plasmids of a flu virus to make it harmless in humans.
Prions, on the other hand, tend to modify themselves into a malformed version since its proteins cannot produce or replicate themselves. In 1997, Stanley Prusner discovered the existence of prions by conducting an experiment to transmit infectious agents into hamsters, and found the brain cells of hamsters were sensitive to the enzymes which digest proteins, but had no effect by the agents which destroy nucleic acid. This led to the belief that such diseases may not contain nucleic acids. A mutation of the normal cellular prion protein, PRPC, causes the formation of the scrappy protein, PRPSC, which is a misfolded protein. This scrappy protein is then able to bind to the PRPC, which induces the protein to fold into the known infectious abnormal conformation. This structure allows the protein to replicate itself and infect its prey, comparable to how rapidly cancer cells are able to replicate.
Both viroids and prions are infectious but differ from viruses as they are much smaller subviral particles that interact with living cells. In order to attempt to stop the transmitting of spindle tuber disease caused by viroids in potatoes into tomato plants, William B. Raymer and Muriel O'Brien had come up with the bioassay. Although their plan was unsuccessful, they continued their plan to rid the spread of viroids among plants. BSC, bovine spongiform encephalopathy, is the scientific name for a commonly known prion disease, mad cow disease. The name derived from how prions infected cows' brains and left only sponge-like holes, causing the cows to behave erratically. Since its discovery in 1986 in the United Kingdom, approximately 200,000 cattle have been affected, although the outbreak has subsided drastically. Unfortunately, BSC left a human version of the contagion in its wake called Creutzfeldt-Jakob disease. It has believed to cross species by the consumption of infected beef. In the 1950s, Carlton Gajdusek became involved in the studies of a neurological disease called Croup, which has only affected a certain group of people, as well as another neurological disease called scrapie within sheep. He later discovered that prions were the agent causing the disease. In the 1970s, those suffering from CJD were treated with human growth hormones from the pituitary gland of human cadavers. Hormone then became available through genetic engineering, which ended the outbreak of CJD.
Prion diseases negatively impact society as they are not illnesses that can be easily treated due to the fact that they generally affect the brain, notwithstanding the fact that they are capable of being sporadic, hereditary, or acquired infection, meaning any person can be diagnosed with prions at any time.
Viroids may cause economic issues because diseases in plants cause for a decrease in sales by the customer and loss by the producer. Moving forward, it would take more money and time to begin disease control on plants, fruits, and vegetables.
Though prions are extremely harmful to mammals, yeast cells are able to withstand and possibly even use the mutation to its advantage. The mutated prion only changes the yeast cell's functioning ability, so if the normal protein acts like a repressor protein, this could potentially allow other genes to activate and produce other proteins that might not have been able to be translated. This could ultimately lead to easy and rapid adaptation to extreme environmental changes if controlled. Furthermore, if prion inheritance is able to be harnessed and manipulated, it would be incredibly advantageous compared to DNA inheritance because of its reversibility. Since DNA is the genetic code, it cannot be changed after mutation. But with prions, it only alters the protein, leaving the genetic code unharmed. In addition, prions are heat and radiation resistant and have the ability to destroy DNA, bacteria, and viral infections. Even better, if the genetic code has the ability to compose a protein such as a chaperonin, whose function is to unfold misfolded proteins, then the mutated prion could easily be refolded properly. Technology on that level would be evolutionary for humans because treating diseases and even adaptation would become simple. Likewise, humans could use prions to prolong lives of patients with orphan diseases and maybe potentially advance evolution, benefiting society and the human race as a whole.